All right, good morning, everybody. It's good to see you again. Today we're going to have a lecture where we're going to be talking about some relatively basic chemistry and chemical principles that will help us explain some very important properties of cells. Does anyone know what the lecture is going to be about today?
pH.
Well, it's not going to be just pH, yeah? Correct. So we'll be talking about the composition of the intracellular fluid, and mainly we will contrast it with the composition of the extracellular fluid, and we'll see how the differences in the composition between these two fluids give rise to some phenomena that we'll be using later on in cellular physiology and cellular biology, etc. We will talk a little bit about pH, but mostly in relation to buffers. So we'll be mostly talking about buffering systems in our body, both intracellularly and extracellularly, and we'll discuss a little bit the theory of buffers as well. But we will talk about more things than that, but that's kind of the basic idea. So if we want to talk about the composition of our cells, of our body, what would you say is the major component of our body?
Water.
It's water. And usually when we talk about composition of stuff, people forget that water is the main component, in which all the other components are dissolved or suspended or whatever, depending on what kind of components we're talking about. Now, what would you say is the proportion that water makes out of a human body? Okay, 60%, 70%. Okay, so first of all, it varies, right? It varies. Hello? Okay, come in and sit down, okay? Right, so it varies and it changes. That's fine, either do it before the lecture or after the lecture, okay? Just don't do it during the lecture. It's really difficult to keep, going. Yeah, you will understand it. Also for people who are coming late, okay, it's really annoying for everybody involved, okay? All right, so it changes throughout life. So newborns have a much higher proportion of water and they're closer to 70% water. But as we age, the percentage of water goes down. And it also depends on the composition of the body. So for example, adipose tissue, fat, contains a lot less water. So people who have more fat, which often is older people, will have lower proportion of water. And people who are leaner, so muscles, for example, contain a bit more water, so they would have a higher proportion. So the figure that you gave, many of you gave 60%, 60, 50%, depending on the age, also on the gender, or biological sex rather. So women tend to have a little bit less water, which is related to the composition of the body, so the composition of the tissues and the proportions of tissues. Okay, so about 50 to 60% of an adult human body is made of water. Now, where is this water? So this water is going to be present in several different spaces in our body. And we can divide this total body water, which we said is about 50 to 60%. So total body water. Okay, 50 to 60% body weight. We can further divide into water which is in cells and which is outside of cells. So we'll be talking about intracellular water and intracellular fluid, which is not just the water, but water containing all the other components of the intracellular fluid. And we'll be talking about the extracellular fluid, which is outside of cells. Now, what would you say is the proportion between the intracellular and extracellular water? Let's put it that way. What could be the ratio, or, so we take the total body water, what proportion of that is going to be intracellular, and what proportion is going to be extracellular? Just guess, if you don't know, just, yeah? 70-30, okay. Any other guesses? 50-50. Okay, 50-50. Any other guesses? Okay, so it's about 60 to 40, so you were all quite close, okay, approximately 60 to 40, so approximately 60% of total body water is going to be in cells, intracellular, and the rest of it is going to be extracellular, okay? So approximately 60% of that is intracellular, and approximately 40% is going to be extracellular. You can see slightly different proportions. Again, it varies with body composition, etc. But those are kind of the ballpark numbers that are worth knowing about. Good. So with intracellular fluid, we can say that it's relatively simple. For now, we'll be thinking about cells as being just sacs of intracellular fluid. Of course, that is not true, and in a second, I'll show you how much it's not true. But we don't really, for most purposes in medicine, we don't really subdivide the intracellular fluid. Just say it's one fluid. Again, that's not true. There are different compartments, and we'll see that. But let's say. But for the extracellular fluid, there is one major division for the extracellular fluid. And that is the division into intravascular fluid. Now, what does that mean, intravascular? Sorry? Somebody, what is it, yeah? The vessel. Correct, in the vessels, and usually we mean in blood vessels, okay? So in lymph vessels, obviously there's some fluid as well. Mostly we're talking about blood vessels, but yes, it is the fluid which we have in blood and let's say lymph vessels, okay? So that's the intravascular. And then we have something which is not called extravascular, even though we could call it that as well, but we call the rest of the extracellular fluid we call interstitial fluid. And interstitial just means between stuff, right? So that's the fluid that we have basically surrounding cells and all other things, which is not in blood vessels, but is around cells, okay? So we have intravascular fluid and interstitial fluid. Now, if you want to have the approximate ratios, again, the intravascular fluid is the smallest one, so it will be about 5% of body weight, approximately, and the interstitial fluid is about 15%, approximately 15% of body weight. Okay, so this gives you approximate proportions of what the amount of fluid we have in these different compartments. Okay, so in medicine, the majority of analysis, etc., will be dealing with the intravascular fluid, right? Most of our analyses, our lab analyses, we take blood from patients or plasma or whatever, some components of the blood. Just one second, and we do all our analysis there. But as you can see, it is just a very small fraction of all the body water, right? And that's usually good to keep in mind, because then changes in the intravascular fluid and the composition of the intravascular fluid may not reflect actually the composition of all the other fluids, okay? So this is, hopefully these proportions tell you that what we usually see in medicine is just a tiny proportion of the reality.
The 5% and 15% is out of the 40?
Out of the body weight. No, out of the body weight. Okay, so here this was out of the 50%. Yeah, I can see how confusing that could be. Okay, but this is actually out of body weight. Okay, so this is this.
60 and the 40 is not.
Those are out of this 50 or 60%. Okay, we can recalculate it. We can easily recalculate them. Okay, those are out of body weight, hence the BW as body weight. Okay. So interstitial fluid is a fluid which is between cells. It's not in blood vessels or lymph vessels. It's just kind of flowing around cells. It's in the tissue, okay? And interstitial just means in between, right? It's in between stuff, okay? So basically that's the rest of the fluid, right? It's either in cells or in blood vessels, and the rest of it is just flowing around, okay? Good. So these are the proportions of water, but they're also the proportions of volume, okay? So these two are related, so you can take that as the amount of fluid that we have in our body. Now, before we look into the detailed composition of intracellular and extracellular fluid and contrast the two, because there are quite big differences, let me show you a picture, well, a video actually, that hopefully will put in your head a slightly different view of the intracellular fluid, what it actually looks like. As I said, usually when we think about cells, and when you, in a textbook, when you look at a picture of a cell, you will see that it's a sac filled with some fluid and there are some organelles flowing around. Well, recently, a few years ago, one research group basically tried to, why doesn't it work? There we go. So one research group tried to make a 3D model of a prokaryotic cell. It's actually a bacterium. So you will see that there are no membrane-bound organelles, okay? But still, it gives a pretty good picture, if we can see something, a pretty good picture of what the inside of a cell actually looks like, and that the intracellular, okay, it's coming up. And that the intracellular fluid is actually really, really tightly packed. Let me just. Really tightly packed with lots of macromolecules, which make it actually into a sort of a gel rather than just a fluid, basically an aqueous solution. So I'll start the video. So here we're looking at the membrane of a very, very small bacterium. It's called Mycoplasma. It's one of the smallest bacteria that exist, okay? So it's a tiny, tiny, that's why they could make it, because for an eukaryotic cell, it would be a really, really hard thing to make the model. So we're looking now at the membrane, and then the membrane will start going away, and this is the cytosol, this is the cytoplasm of the bacterium. And you can see that it's filled, really packed with different proteins. Now the yellow ribbons, that's the DNA. Okay, again, in bacteria, we don't have a nucleus where all the DNA would be stored. Okay, so it's just kind of distributed in the cytosol. But you can see that really all the different enzymes and structural proteins, etc., are packed really, really closely to each other. Okay, and in fact, we'll see in a second the actual composition. We'll see that the amount of protein in the intracellular fluid is similar to the amount of protein in egg white. Okay, so imagine, or think about the texture or the viscosity of an egg white, uncooked egg white, and that's what the inside of our cells actually looks like. So it's sort of between a gel and really viscous fluid. Okay, it's not really just an aqueous solution. So hopefully this model gives you a slightly different perspective on what the inside of our cells look like. All right, let me switch this off. Let's see if it works. Maybe not. Good. And let's get the lights on. That's better. Good. So now let's talk about the different components in the intracellular and extracellular fluid, and let's see the differences between them. So one of the the major component is, as we said, is water in both of these types of fluids. And then let's start talking about the components, starting with electrolytes. So electrolytes is just a word that is often used. What does it mean, electrolytes? Salts, yeah, they could be salts. Ions. I think that the best equivalent is ions, okay? So all sorts of ions that we have dissolved in all these fluids, we often in medicine call them electrolytes because they obviously conduct electricity, etc. But the correct name would be ions, okay? So we have water, we talked about that. And let's now talk about the ionic composition, what type of ions we have. So we will not, at this point, talk about these macromolecules, these large proteins, even though, proteins and DNA, etc., even though, and we'll see that later, there are also ions. Okay, so even these macromolecules actually act as ions because many of these macromolecules they have groups that can dissociate, okay, either release a proton or accept a proton. So we'll get to those as well. But let's start with the small ions because that's where we will see the major differences. Now, how to have a kind of a visual way to see the differences. So let us get columns like this for cations and anions, and we'll see that for the intracellular fluid and the extracellular fluid as well. So let's start with the extracellular fluid. And we have cations and anions. Okay, and we'll do the same for the intracellular fluid in a second. Because this gives us nicely the differences and the proportions, which are maybe not as easy to see from numbers themselves. Okay, is it clear what we have here? Extracellular fluid, cations, anions. All right, so what is the major cation in the extracellular fluid? Does anyone know? It's sodium. Okay, so the major cation in the extracellular fluid is sodium, and it's not going to be exact, but does anyone know what approximately the concentration of sodium is in the extracellular fluid? Approximately? Not in percent, but actual concentration, okay, because it would be hard to see what are we taking the percentage from. Sorry, somebody said something. No? Okay. Well, so yeah, okay. Maybe you're asking what kind of concentration are we going to use for this, okay? So we're going to use molar concentration. That's usually the most useful concentration, and that's the concentration that you'll see in your patient's lab results. Most of the concentrations, at least here in Europe, you will see them in molar concentration. So the molar concentration of sodium in the extracellular fluid is approximately 140 millimolar, okay? Approximately 140 millimolar. And this is something, I mean, I'm going to give you approximate numbers for all the ions. At this point, you should at least learn these approximate ones, okay? So when somebody asks you how much sodium in the extracellular fluid, you're going to say 140 millimolar, okay? There are exact ranges, and at some point, we will expect you to learn the exact ranges, the exact normal ranges, which are quite important because then you can tell if a patient has too low sodium or too high sodium, etc. But at this point, from this lecture, it's enough if you remember these ballpark numbers because they give you the proportions and the differences, okay? So approximately 140 millimolar sodium. Now, if we look at the intracellular fluid, again, cations, anions, intracellular fluid, does anyone know what the major cation is there? It's not calcium, it's potassium, okay? So the major cation in the intracellular fluid is potassium, and again, if you remember as a ballpark number 140, it's approximately right, 140 millimolar. Okay, so that's our first big contrast between the intracellular fluid and the extracellular fluid. Now, what are the rest of the the other major cations in the extracellular fluid? Well, the next one, and here I will stop asking you because I understand it's going to be harder and harder to guess. The next one in the extracellular fluid is actually potassium, and as you would expect, the next major cation in the intracellular fluid is sodium, but the amount of sodium is much smaller. So in the extracellular fluid, the concentration of potassium is approximately 4 millimolar. Okay. Let me write it somewhere. About 4 millimolar. Remember the units, okay? Never say a concentration or a value without the units because then it doesn't mean anything. If you just say 4, what does it mean, right? 4 millimolar. And the concentration of sodium in the intracellular fluid is about 10, 12 millimolar, thereabouts. Now, let me just give you one caveat, one kind of note of caution. While the concentrations in the extracellular fluid are actually really well known and easy to measure, as you can imagine, because you just draw some blood and measure the concentrations in the intracellular fluid are much harder to actually measure accurately. Okay, so whenever you look into literature, there's going to be a very strict agreement with the values of ions and other components in the extracellular fluid. For the intracellular fluid, you can find quite different values because they differ in different cell types, they differ in different situations, etc., etc. So they're not that easy to measure. Just be aware, because if you start looking at other materials, you might see different values. They're not going to be widely different, but they can be quite significantly different. All right. The remaining two main cations in the extracellular fluid are calcium and magnesium. And the ballpark values that you can have is about 2 and 1 millimolar. Again, ballpark, okay? At some point we'll need the exact ranges, but here this is enough. For the intracellular fluid, the next big one is actually magnesium. And calcium, and we'll talk about calcium quite a bit, the intracellular calcium is actually very, very low. The concentration is tiny, okay? It's about 100 nanomolar or something like that. It's very, very small. And let me just give you kind of the first inkling of the importance of calcium, because we'll talk about it more in the third course in the beginning of the summer semester, because intracellular calcium is very important for signaling. So if the intracellular calcium goes up, usually the cell will do something, okay? A muscle cell will contract, or a glandular cell will start excreting something, etc. So the concentration of calcium in normal cells is kept very, very, very low, okay? Unlike the extracellular. So the extracellular calcium is quite high. Intracellularly, it's tiny, tiny, okay? Unless the cell is supposed to do something. Yeah, and the magnesium, the concentration of magnesium is about 7 millimolar. There was somebody who wanted to ask something? No, right. Okay, let us have a look now at anions, okay? The major anion of the extracellular fluid, okay, so blood, but not just the blood, is chloride. Yeah, absolutely. It's chlorides, okay? And the concentration is approximately 100 millimolar, okay? So it's about 100 millimolar. Now, the major anion in the intracellular fluid, inside cells, is a bit tricky because it is actually phosphate groups. But before you start writing phosphate, we have two different kinds of phosphates, let's put it that way, both in cells and outside of cells. One is inorganic phosphate, so the stuff that you know as hydrogen phosphate, dihydrogen phosphate, etc., right? It's just inorganic phosphate, and there is some there. The other type is organic phosphate. Now, what is organic phosphate? Can anyone tell me what that might be?
Phosphates are organic molecules like ATP.
Correct. So usually there are esters or some other derivatives of phosphoric acid, but they are organic compounds. So it could be ATP, it could be all sorts of phosphorylated proteins, etc., etc. It could be DNA, okay, etc. So these are organic phosphates. Now, the major cation in the intracellular fluid are these organic phosphates, okay? The concentration, the estimated concentration, let me just have a look, is again about approximately 100 millimolar. Organic phosphates, okay? Now, why am I telling you all these details and kind of pointing out that they're organic phosphates? Because again, in literature, these two get often confused, and they will just tell you, well, there is a 100 millimolar phosphate concentration. But that's not actually true. The concentration of inorganic phosphate inside cells is actually quite low. It's between 2 to about 7 millimolar. Okay, so this would be the inorganic phosphate. For the inorganic phosphate, we usually abbreviate as PI. Okay, PI, it's inorganic phosphate. Can you read it? PI. P subscript I. Let me. So Pi is about 2 to 7 millimolar. Okay, so inorganic phosphate is actually relatively low, but if we take all the phosphates together, both the organic and inorganic, they are by far the major anion in the intracellular fluid. Now, the second biggest anion in the extracellular fluid in the blood is bicarbonate or hydrogen carbonate. And we'll talk quite a bit more about it today because it's one of the major buffers in the blood. Okay, so this would be the second most abundant anion in the extracellular fluid. We have about 25 millimolar concentration of bicarbonate. In the cells, in the intracellular fluid, the second most abundant anion are proteins. And I'm going to put a minus sign next to proteins, because as you know from the structure of proteins, they can have both positive charges and negative charges, depending on what? What will determine what kind of charge the... Yeah, correct. So the composition of the amino acids in the chain. So of course, we have proteins that will primarily be positively charged, but the majority of proteins will be overall negatively charged. Okay? So most proteins will work as negative charges. Now, the estimated concentration of protein anions in the cell is about 40 millimolar. 40 millimolar, approximately. Now, let me complete the intracellular fluid, and then I'll come back to the anions of the extracellular fluid because there's something else that we need to kind of distinguish, okay? So the remaining anions of the intracellular fluid are bicarbonate, and we already talked about the inorganic phosphates, so there's going to be bicarbonate. The intracellular concentration of bicarbonate is, again, fairly low, about 10 millimolar, and the rest are chlorides with about 3 millimolar or thereabouts. Okay, so inside cells, the majority are organic compounds, organic anions, and then we have a few of the inorganic ones. Now, let's go back to the extracellular fluid, and now we have to start distinguishing between the intravascular fluid and the interstitial fluid, because here we're coming to another difference between these two. So now we're talking just about the extracellular fluid. We have to distinguish the intravascular and the extravascular. And the main difference between these two is in the concentration of... Well, yes, but which ions? Proteins, actually. So, as you can imagine, or as you know from your other studies, in the blood we have quite a lot of protein, right? In the plasma, there's albumin, there are all sorts of other proteins, but in the interstitial fluid, in the fluid which is around cells, the concentration of proteins is much, much, much smaller. Okay? So the main difference, and again, we'll use this difference later on to talk about some osmotic pressures, etc., etc. It's very important for the functioning of circulation, especially microcirculation. So here we need to distinguish between the intravascular fluid and the extravascular fluid, or the interstitial fluid. So if we are talking about the plasma, blood plasma, so the intravascular fluid, we will have the next component, the next big anions are going to be proteins with a concentration approximately 14, I think. 14 millimolar or thereabouts. Okay, so that's in the blood, that's in the plasma. Okay, but in the interstitial fluid, the concentration of proteins is so small that it wouldn't be a major anion at all. Okay, so the concentration is much, much, much smaller, something like 1 millimolar or even less. Okay, so it would not actually be a major anion there. Finally, we will have some inorganic phosphates in both the interstitial fluid and the intravascular fluid. And the rest are some other anions. And later on, when we talk about metabolism, etc., we will talk about some organic acids, which can be found in the extravascular fluid, but also in the intravascular fluid, which are the product of metabolism, and they also act as anions. So this would be other. Okay, I didn't tell you about the extracellular phosphate. It's about 2 millimolar. So, hopefully, you can see the main differences between the intracellular and the extracellular fluid. There is one difference with proteins also between the intravascular and the interstitial fluid, or the extravascular fluid, right? Otherwise, the rest of the concentrations are very similar between the intravascular and the interstitial fluid. Good. Now, it's probably not going to happen if we add all those numbers now together because they're approximate, but the important thing about the composition of all these fluids is that they are electroneutral. What does it mean that they are electroneutral? The sum of all the anions is equal to the sum of all the cations. Now this is quite important because, and you may know that from physics, even very tiny differences in composition or the ratio or between cations and anions will cause really huge electric fields. Okay? So in our body, if there was disagreement between the amount of cations and the amount of anions in our cells, the cells would be electrically charged, which they are, as we'll see in a second, okay? But basically, overall, the concentrations should add up. Now, again, because I gave you just some ballpark numbers, they may not exactly add up, but inside cells and outside of cells, the number or the concentration of anions and cations have to add to zero or subtract to zero, whatever. They have to be the same. Otherwise, you would get massive charges, massive electric charges, okay? So both the inside of the cells and the outside of the cells is basically electroneutral, which is helpful because then if you need to calculate something, you can always say, okay, the two should be the same, so I can kind of add or subtract to get the same numbers. So, if we measure concentrations, we'll get the same number of cations and anions. However, there is an important phenomenon in our body, in some cells, in most cells, and that is that many cells are actually electrically charged, that they have a membrane potential. So they have a difference in the amount of positive charges and negative charges across their plasma membrane. Now, how is that possible? How does it come about? Okay, if we said that the amount of cations and anions are basically the same, both inside the cells and outside of cells, how does it come about that some cells have membrane potentials, that if you take a cell and measure the potential, the electric potential between the inside and the outside of the cell, you get some number, okay? And the number is not small, okay? So for example, neurons will have the membrane potential of about -60 to -70 millivolts, which is actually a very large charge because the membrane is really thin, right? So it's a massive electric field. So how does it come about? The main principle that guides this formation of this membrane potential is not the difference between the positive and negative charges inside and outside. I mean, it sort of does, but it has to be created. But the main thing, the main principle for creating this membrane potential is the difference in the composition of those ions between the outside and the inside. I'll say that again. It's not that the cells just when they, come about, that they will have different amounts of positive and negative charges inside. They will get there, but that's not the beginning of it. The principle that creates potential is the difference in the composition of especially the cations. how does it work? Well, so we have intracellular fluid and extracellular fluid, right, as we just said. And without anything else happening, as we said, these would be electroneutral. There would be no charge, okay? Because the sum of positive negative charges inside, the sum of positive negative charges outside are the same. Now imagine that you introduce, so this is the plasma membrane, imagine that you introduce into the plasma membrane a hole or a channel that only allows potassium ions to go through. Okay? So there's a hole that only allows potassium to go through. What's going to happen? Based on the stuff that we... Huh? What will become positive? With respect to what?
Potassium will get out of the cell?
So, okay, lots of different ideas.
Yeah, potassium will leave the cell.
Right, so let's start with what will actually happen with the ions. So you're saying the potassium will start leaving the cell. Why? Okay, it's not osmosis. We'll get to osmosis. It's not osmosis, but it's a different word for that. Diffusion. Okay, so the process that happens is diffusion. Basically, the potassium ions that are inside and the potassium ions that are outside will just start diffusing because there's a hole that allows them to go through, right? So some potassium ions will go in, some potassium ions will go out, right? Diffusion is just a random process. The ions just move around, okay? They don't know where they're going. So some people mention gradient. What is gradient? Difference in concentration. So what does a gradient have to do with this? Again, diffusion is just a random process, right? The ions just kind of move in the solution, and they can move in or out. Why do they move from the higher concentration to the lower concentration? How do they know what concentration is around them? It's magic, isn't it?
They want to achieve equilibrium.
They want to achieve. So you think this tiny potassium ion is thinking, oh, I really want to achieve equilibrium. That's the same thing as equilibrium, but how do the ions know which way is towards equilibrium? It's not osmosis. It's related to osmosis, but it's not osmosis. The process we're looking at is not osmosis, it's just diffusion. It's closely related, and we'll talk about osmosis in a second, but that's not what it is.
There's a higher concentration of potassium ions inside, so there's just a higher chance of them going outside.
Correct. Yes. So the ions are moving completely randomly. They have no idea that they are in a space where there is a higher or lower concentration. They don't know that. But statistically speaking, there is a higher chance that a potassium ion will go from the inside out, because there are more potassium ions in than out, then a potassium ion from the outside will go in, right? That's it. It's just a statistical process, okay? We'll talk a little bit later on in the second course, we'll talk about thermodynamics, and there are some principles of thermodynamics that kind of describe this. But basically, all we need to know is that the ions are moving randomly, but since there are more of them and significantly more of them inside than outside, overall the net flow is going to be from the inside to the outside, right? Just by random movement. Okay? Good. So potassium ions overall, I mean, they're moving both ways, but overall they're moving outside, right? More of them are moving outside. So what will this cause to the electroneutrality of the cell? It will stop being electroneutral, right? Because charges are moving out. Correct. So the inside of the cell will become negatively charged, and the outside of the cell will become positively charged. And this is what we see. So by convention, the membrane potential is taken as inside with respect to outside. That's why we write minus 60 millivolts, because it's negative inside. Okay, that's the convention. We use this. You could do it the other way around, okay? But this is how it works. So it's negative inside. And this is the sufficient process to create resting membrane potential in a cell. So when you read about membrane potential establishment or whatever you hear about the sodium-potassium pump and what have you, okay, these all take a little bit of part in creating and maintaining the potential, but this is what actually creates membrane potential in all our cells that have membrane potential. It's the permeability to potassium. And you don't really need anything else to create a potential. Okay? So in muscle cells, in nerve cells, in cardiac muscle cells, etc., this is the process that will create the resting potential. Okay?
Is there difference in concentration between the potassium to the potassium, or between all the concentration from the inside cellular?
Yeah, that's a good question. So what determines the outflow or the flow of potassium? Is it just the difference in the concentration of potassium, or is it all the other differences as well? Does anyone want to answer that?
When you said about the resting potential.
Sorry, are you answering the question or?
No.
Okay, so I said, does somebody want to answer that question? Whether the outflow or the net flow of potassium is determined only by the concentration of potassium or by all the other ions as well?
By the other ion as well?
Not really. So if the only permeability that we have is permeability to potassium, the only thing that will cause the net outflow of potassium is the difference in the concentration of potassium. Okay? So if this model is correct, we only have permeability to potassium, the only thing that matters is the concentration of potassium in and out. Of course, once we start adding other permeabilities to chlorides and whatever, then they will play a role as well. Okay? But if there is no permeability to all the other ones, they don't matter at all. Okay?
When you were saying that like the cell membrane potential depends on the movement of potassium, is that like period end of sentence, or were you just using potassium as an example?
No, that's how it actually works.
Okay.
It's actually almost all potassium. Okay? In real cells, we do have other permeabilities, so there's permeability to chlorides and there's permeability to other things. So the exact amount, the exact number for the potential, actually, I will tell you something that normally I talk about in the third course, okay? But if you just let this keep going, okay, when will it stop? It won't stop. Sure, there will be in and out, but is there going to be a net flow all the time? It will just never stop? When will it stop? At equilibrium. Now, what is equilibrium in this case?
When the outside and the inside potassium are the same.
Okay, so one suggestion is that this process will stop once the concentrations outside and inside are equal. That's not the case, actually, okay? Because that would not work, okay? But that's not the case, and I'll explain why. The reason it will not work is, and I'm coming back to this idea that even small movements of charges, small differences in charges, cause huge electric fields. So what will happen here is that the potassium ions will start moving out, the inside will become more and more negative, and very early on, even with just a few thousand ions moving out, the electric field will become so high that it will start pulling the potassium back in. Okay?
It just keeps going, like outside and inside.
Well, yes, like at the level of individual ions, it will start going, it will just keep going all the time, okay, in and out. The concentrations will not become equal, no. They will actually stop, as I said, just a few thousand or maybe tens of thousands of ions. It's a tiny amount, okay? We can't even see it in the concentrations. You will not see any difference in concentration. It's just so tiny. But this will cause such a huge electric field that it will start pulling the potassium ions back in and will reach an equilibrium. But the equilibrium is between concentration difference, concentration gradient, and electric field. And we'll reach this equilibrium, which is actually at minus 90 millivolts. So it would reach a potential of minus 90 millivolts. Okay, and basically stop. So the net flow of potassium will stop. We don't really see this high potential because we have other permeabilities there. So this minus 60 in a nerve cell is caused by the fact that there are other ion channels, so the actual potential is lower. But in theory, we would get minus 90 millivolts and the outflow will stop. You wanted to ask something? No? You wanted? No? Somebody wanted? No? Good. Any questions about electrolytes or ion composition and the formation of the resting potential? Yes?
What is the significance of the potential electric charge?
What is the significance of it? Well, many cells that have this membrane potential are capable of changing this potential, of depolarizing their membrane. And this is a very important signaling method. For example, in our nerve cells or muscle cells, etc., it's the change in the potential which causes the cell to contract or whatever, okay? And we'll talk about that in third course quite a bit in signaling. Here it is just establishing how we get the resting potential, okay? And then we'll talk about the changes in potential and depolarization and action potential and or what have you.
The same potential that our body uses to contract muscles?
Correct. It's the same potential, or rather the changes in the potential are what contracts muscles and etc. Just speak up a little bit. So, okay, these are quite specific questions about how gated these ion channels are. Mostly they are voltage gated, but let's not worry about that. Let's just imagine that they are open all the time. Okay, let's just imagine that they are there and without any gating at all. Okay.
When you talked about the resting potential and you said minus 90,
Yes.
then you also said that it's barely ever happens.
Correct.
So it's only like in theory to understand that in minus 90 it will happen, but
Yes.
it never happens.
In our cells, we never get to minus 90, or almost never get. I mean, I don't want to go into too much detail. Sometimes we get to minus 90, but mostly we don't. But the reason for that is that in our normal cells, we don't have just permeability to potassium. We also have other ion channels, and they will change the equilibrium potential because there are some chloride channels or calcium channels and what have you.
So this is just the resting potential of the potassium.
This would be the resting potential if only potassium permeability was there, okay? But in our normal cells, that's never the case.
If you add more stuff, then the resting potential the number will change.
If you add other ion channels, it will change, yes. Yep. Good. Any other questions about this composition and the potential? Yeah.
Isn't the minus 90 the hyperpotential between the resting state?
Yes, it is, and that's what I really didn't want to go into, because then we'll be talking about action potential and it's a bit complicated. But this is what we call the equilibrium potential for potassium, and basically we can't get any more negative than this. So there are situations when the cells can go all the way to 90, but they can't really go any more negative, okay? So at a resting state, we don't have cells with minus 90, okay? But there are situations where they do overshoot to minus 90. Yes?
So there's barely a change of difference in concentration
Correct.
of potassium, but there's a very deep change in the...
Correct, exactly, okay? So literally when I said that only a few thousand or tens of thousands of ions, which is a tiny, tiny, tiny amount in the cell, need to exchange place in order to get to this potential, okay? So by measuring concentration, we see nothing. There's no change in concentration because it's a really, really tiny amount.
This minus 90 has come because we've taken the reference point from inside to outside, but hypothetically if we take it from outside to inside.
Sure, it will be flipped, yeah. But the convention is that we measure inside first with respect to outside, yeah. Okay, let's take a 3-minute break, but literally 3 minutes, okay? So go to the loo, drink something, and let's come back and we have still a few things to talk about. All right, so when I talked about the formation of a membrane potential, you saw that the main reason, the main mechanism for the formation of membrane potential, of electrical membrane potential, is the difference in ion composition connected to or augmented to or activated by selective permeability of the membrane. So the membrane in most cells at rest is selectively permeable just to potassium, and that will create the resting potential. If the membrane was permeable to everything, we wouldn't get any membrane potential because some ions will be flowing in, some ions flowing out, and it would basically end by just equalizing the concentration. That's it, okay? So we have selective permeability, and that's what enables the formation of the resting potential. Now, continuing with this idea of selective permeability, we can now talk about another related phenomenon, but different, and one that many of you mentioned already, and that's osmosis. So osmosis is a phenomenon, and many of you, most of you, have heard about it and have probably heard of some definition. But osmosis is a process where a solvent, usually water, because we're working with biological systems, so it's usually water, doesn't have to be water, but usually it's water, that a solvent is able to move through a semi-permeable membrane. I'll say that again. In this example with membrane potential, we saw that a membrane is permeable just to potassium. Now imagine that you have a membrane, and actually I can do it as a cell as well. So you have a membrane, but this time it is permeable only to water. So it has channels which allow water molecules to go through, but nothing else. Now, if the composition of the, in this case, intracellular fluid and extracellular fluid is the same, and I'll mention in a second what it means to be the same, the water molecules will just move in and out, but the net flow is going to be 0, right? What kind of the same concentration do we have to have, or the same, the composition rather, not concentration, in order for the water molecules to just flow in and out without any net flow in or out? Does the question make sense? No. Okay. So I said, well, previously we looked at the differences in concentration of potassium, and we said that they are different, and that means that if you open a channel for potassium, the potassium will flow out because there's a difference in concentrations. Here we're talking about water molecules. So what aspect of the composition in and out has to be the same in order for the inflow and outflow of water to be essentially 0?
H plus and OH minus.
It's not H plus OH minus.
Concentration of water?
Correct. Yes. Concentration of water. So, if the concentration of water inside is the same as concentration of water outside, there will be no net flow of water. So water molecules will go in and out, but there will be no net flow, right? But if the concentration of water is different, we will get a net flow of water either in or out, okay? It's exactly the same thing as with the potassium gradient, okay? This is just water concentration gradient, okay? So we're talking about the same thing. Again, it's just a random statistical process, okay? But if there is more water inside than outside, there will be a net outflow of water, okay? And if there is more water outside, more water concentration, not amount, concentration of water, right? If there's more water concentration outside, then water will flow inside. And that's osmosis, okay? That's all there is to it. Okay? It's really just about the concentration or the differences in concentration of water.
Doesn't water also follow sodium molecules in and out of the cell?
Yes, but that's just the implication of what I just said. Okay? So let me use this as an example of how we can change these concentrations. So imagine that once again we have a cell, okay, and the concentration of water inside is the same as the concentration of water outside. And we have a channel which allows water molecules to go in and out. What happens? Nothing, right? Because the concentrations are the same. Now, let us imagine that we have a transporter or a channel that allows one sodium atom to move from the inside to the outside. Okay? So one sodium atom, one sodium ion has been moved out. Does this in any way change the concentration of water?
Yes.
How?
Because it makes water more dilute, so there's more water molecules
Where? Where is going to be more water?
in the cell. If sodium leaves, then there's more water in the cell.
Correct, right? We removed one atom of a solute. So that means now we have relatively more water here than here. Therefore, we get a difference. There is now more water inside than outside, and therefore, water will flow out. And that is how water follows sodium. It doesn't literally, like, you know, hitchhike and it kind of catches the iron. No, it doesn't, okay? But by moving solutes, ions, glucose, whatever, across the membrane will change the concentration of water, and this will cause a net movement of water either in or out, depending on where you're moving the solute. Now, very important thing. In the case of membrane potential, we only talked about charged species, about electrolytes, about ions, okay? Because only ions can carry electric charge. So we can't get electric potential by moving non-charged atoms, right? That makes sense. In the case of osmosis, that is not the case. Even if we take, for example, a molecule of glucose, which is not charged, and move it from the inside outside to the outside, we also change the concentration of water because previously glucose was dissolved in this water, right? So one molecule of glucose moves out, that changes the concentration of water and will cause osmotic movement of water, right? So for osmosis, it's not only about ions. Actually, any molecule is what we call osmotically active species, osmotically active molecule. Okay, because often this is confused, okay? Often people think that only ions will cause osmosis. That's not the case, okay? Any kind of molecule which is dissolved, so, you know, a chair is not going to cause osmosis, right? If you throw it in water, it's not going to be dissolved, right? So it's not going to cause osmosis. But anything that is dissolved and you move it or you get a change in concentration, you will get osmosis if there is a permeability for water. So I'll summarize it and then you can ask questions, okay? Osmosis is a process, is a phenomenon where the molecules of a solvent, again, in biology it's water, okay, move across a semi-permeable membrane. Semi-permeable, well, because it is only permeable to water. If it is also permeable to sodium, right, the sodium will just flow back in and nothing happens, right? So that's why I said it's a transporter or something that moves the sodium out, okay? Or there must be a concentration gradient, there must be something else that moves it around. But if it's a semi-permeable membrane that only allows water to flow in and out, if we change the concentration of dissolved particles, of dissolved molecules, all of them, charged, uncharged, ions, glucose, whatever, if we get a difference, then water molecules will flow in and out depending on the concentration of water. And this is osmosis. So you can see that it's related to what we talked about with potassium and membrane potential. The biggest difference is that here it's water moving in and out, and any kind of dissolved molecule will be osmotically active. Not just the charged ones, all of them are, because they are changing the concentration of water. Questions? Yes?
You just said that the membrane is only permeable for water, so how can like glucose or sodium?
Yeah, so imagine that we just take a hand and just pull it out, okay? Don't worry about the process, how we change the concentration. I'm just trying to show you that if you change the concentration of solutes, you also change the concentration of the solvent, and that's what causes osmosis. So don't worry how this works, okay?
And like the molecule also like always aspires that the water concentration inside and outside would be the same.
Not just water concentration, okay? If you get any concentration gradient, any concentration difference, just by this statistical random process, there will be a tendency to equalize the concentrations. But in this case, the membrane only allows the through-going of water, right? Nothing else. So even if there is a change in concentration or difference in concentration of sodium, it can't go anywhere because there are no channels for that.
The water will go outside and inside.
Absolutely. But if there's more water inside, the net flow is going to be outside, right?
And if there is too much outside, then it will go.
Then it will go in. Yeah. Any other questions about osmosis?
It's kind of a weird question, but like in plant cells, there are vacuoles.
In what cells? In plant cells. Okay. Mm-hmm.
Is there also like a semi-permeable membrane like around?
Yeah, absolutely. Yeah. Plant vacuoles have a membrane which is permeable to all sorts of things. Yeah, absolutely.
Well, so the osmosis in the plant cell will happen twice, like once between the vacuole and
Absolutely. Yes, it will. Okay. Whenever there is a compartment which has a membrane which is permeable to water, which is almost all, not all, but most membranes, then you will get osmotic pressure changes depending on the composition of the inside and the outside. Yeah.
So suppose one sodium ion is moving from inside to outside, that causes a higher concentration of water inside the cell, and therefore the water will move from a higher concentration.
Absolutely. That's what happens. So 2 things to add. One is, well, what are these channels for water? They really exist, okay? So most our membranes have these channels. They're called aquaporins. Aquaporins, and they are just channels for water. We need them because we need to move water around, okay? So basically they're everywhere, okay? All plasma membranes and many of the membranes in the cell have aquaporins, and we get water moving around, okay? So this is just the mechanism through which water can flow.
Is it special just for sodium ions? Or like
No, that's what I was trying to say. Anything that is dissolved, if we get a difference in the, and I will say that again in a second, okay? Any kind of species which is dissolved, if the concentration changes of those solutes, the concentration of the solvent also changes. And that's what causes osmosis. Let me add to it, okay? So how do we find out, how can we know whether there's going to be a net outflow or a net inflow of water in any kind of situation, okay? Inside a cell, outside of a cell, whatever. Well, what we do is we just sum all the dissolved ions inside and outside, and if there's a difference, okay, water will flow from where there is less dissolved stuff to where there's more dissolved stuff. Okay, let me show you maybe more kind of graphically. Okay, so let's take the inside of the cell. Okay, we had all the components, and we just sum all the concentration of all the stuff. So we sum sodium ions, potassium ions, chlorides, glucose, urea, ATP, everything that is dissolved, okay? And we get a sum of all the particles. And this will be measured in moles per liter, right? So we get some concentration of all the dissolved stuff in moles per liter. Right? And we do the same thing for the outside. We sum all the dissolved particles and we get some concentration in moles per liter. Now, just looking at the difference in the concentration of the dissolved stuff will tell us whether water will flow in or out. Okay? Because where there is higher concentration of dissolved stuff, there is lower concentration of water. And when there is lower concentration of dissolved stuff, there is a higher concentration of water. And water will just follow its concentration. That's all there is to it. Now, this molar concentration of all osmotically active particles is often abbreviated, kind of changed into osmol per liter. It's exactly the same thing, only we're talking about all osmotically active particles. So we're not talking about sodium separately and potassium separately and chloride separately. We just say all the osmotically active particles, we sum them all together, and then we can use these units osmol per liter. But they are just mole per liter, just summed over all the dissolved particles. That's all there is to it, okay? So there are not some mysterious different units. It's just moles per liter summed over all the components. Yes?
So when sodium, for example, wants to leave the cell, does it leave while being dissolved? Like, does it stay dissolved and leave?
I mean, here we would be getting into like too much molecular processes, okay? Sort of, yes, it does, sort of it doesn't. It's very complicated thing what happens inside the channels. I don't really want to go into that, okay? But the osmotic processes usually happen not in like one go, that basically the sodium just pulls one molecule of water with it. Usually, sodium is moved, and then through some other channel, water is moved, okay? It can happen together in certain cases, okay? But in your heads now, it should be a separate thing. So we have channels for sodium, potassium, and then we have channels for water, okay? Just to make it separate and clean. So that's osmosis, okay? You all look a little desperate, but hopefully you will see once you look at it again that it's the same thing. It's just stuff following its gradient, just following its difference in concentration. That's all, okay? Only with osmosis, it's water, and the stuff that changes the concentration of water is just how much stuff there is dissolved in it. That's all there is to it, okay? The final bit that we need to talk about is a little bit of pH. Now, I'm not going to be defining pH because we've done it and we've done it together. I'm not going to tell you how to calculate pH. Again, that's something that you've done and you've seen before, okay? I will just give you a few kind of pointers, and I'll talk a little bit about buffers. You know, how we can get from the theory to buffers and what buffers are and how they are used. So, a few things. We already talked about it in the prep course. Not all of you were there, so let me just repeat. We have different ways of calculating pHs of solutions for strong acids and weak acids, okay? You should know the formulas for both of them. Do you know the formulas for both of them? No, you don't. All right, okay. So for strong acids and strong bases, how does it work? How do we calculate the pH? That's the simple bit. Huh? So, yeah.
You find out the concentration of H plus, and you do negative log of that?
Correct. So, yeah, the definition of pH, again, we did it before, is minus log concentration of H plus in the solution. That's all there is to it, right? In the case of strong acids or strong bases, we basically assume that all the molecules dissolved have dissociated into protons and anions, right? In the case of bases, it works the other way around, but you should know that, hopefully, okay? So in the case of strong acids, we just say everything that we put in, everything dissociated. Therefore, the concentration of hydrogen ions is going to be equal to the concentration of the acid that we put in, right? Because everything dissociated. Now, of course, if the acid can dissociate into more than one proton, then we have to multiply it by the number of protons that are released. But we just assume, for strong acids, we just assume everything is dissociated, okay? For weak acids, that's more complicated because weak acids are not completely dissociated, okay? So we can't just assume that the amount of protons is equal to the concentration of the acid because some of the acid will remain as acid and not as the anion, okay? So how do we distinguish between strong acids and weak acids? Well, yes, but how can you tell that it's full dissociation or not? But we use some number.
PK.
Correct. We use pKa. What is pKa?
The negative log Ka.
It's the negative log Ka, and Ka is the dissociation constant for the acid. Okay. Again, I'm not going to go into that. You should know these things. Okay. You should know this. Okay. So based on the pKa, we can tell approximately if it's going to be a strong acid or a weak acid. Do you know the numbers approximately? So if pKa is less than 0, it's definitely a strong acid. It's a strong acid, okay? If it is more than 3, it's weak acid. In between, that's tricky, okay? It's something in between. Remember in the prep course I told you it's arbitrary, okay? It's a range. So we can't say here this acid is strong and this one is weak, okay? It's a range that changes. So anything smaller than 0 is going to be strong acid, anything larger than 3 is going to be a weak acid, and in between, well, that's tricky, okay? Now, For the calculation, and I'm not going to show you the derivation of that, even though it's relatively easy to derive with just one assumption, but the pH of a weak acid is 1/2 pKa minus 1/2 log concentration of the acid. Okay, and again, that's something you definitely saw and you have in your workbooks, so you can work through the problems, but this is the formula. I'm not going to show how it's derived. It's very easy to find on the internet how it's derived, okay? There's just one simple assumption there. Now, let me just briefly add something to the strength of acids, okay? So we talked about strong acids and weak acids, but actually some acids are so strong in very concentrated solutions that the whole concept of pH actually stops working, okay? So for very strong acids and their concentrated solutions, we can't actually use pH because it doesn't make any sense, okay? We use something called a Hammett acidity function, which is not something you need to know or you will never come across it. But what I just wanted to say is that we have weak acids, strong acids, some in-between acids, but we also have super acids. Again, this is not something we'll come across in medicine, but it's just an interesting fact. Well, it's interesting for me, so I'll tell you. I'm not sure if it's going to be interesting for you. But there exist superacids, and they are defined as acids which are stronger, which are more acidic than concentrated sulfuric acid. Okay? So you have 100% sulfuric acid, and these are stronger acids than that. Okay? They are really, really weird acids, okay? They're usually mixtures of several different compounds, like antimony pentafluoride with hydrofluoric acid. If you mix those two together, you get a super acid, which is so strong that you, for example, can put in propane, okay, just an alkane, which normally doesn't react with anything, and they will add a proton to the propane or to methane or whatever. They will just take a proton and add it to it, and you get something like CH5+. Really weird. They are just so strong that they are capable of giving proton to anything. They are very useful in chemical synthesis and making things. But just so you know, we have strong acids, and then we have super strong acids, which are super interesting. And if you like this kind of thing, you can read up on these. Right, the last bit. So this is just a really quick run through pH. You have plenty of that in your workbook. Work through the problems, and we don't really need to spend a lot of time talking about that. What we do need to spend a bit of time, we have 5 minutes or 6 minutes, but what we do need to spend a bit of time with is buffers or buffer solutions. What is a buffer? Yes?
Solution that can resist a change in pH.
Correct. So it's a solution which can buffer, which can resist changes of pH caused by adding a strong acid or a strong base. Okay? Why do we keep talking about them in biochemistry? Well, because in our body, in our cells, we need them all the time, because our cells are producing mostly acids, but sometimes even bases, but mostly acids, and we need to have systems to regulate the pH, because most of our enzymes and other proteins, signaling proteins, etc., need a very narrow pH range, outside of which they stop working. Okay? So we have these systems, these buffer systems, to regulate, or rather to stabilize the changes in pH inside our cells and also outside of our cells. How do buffers work? Okay, so usually they are composed of a weak acid and its salt, or a weak base and its salt. And the idea is actually relatively simple. So, if we have a mixture of some weak acid and its anion, its salt, right? Then, if we add some proton, this proton will, because it's a weak acid, this proton will have a tendency to bind to the anion and disappear from the solution. Because a weak acid really wants to bind some protons. That's what makes it weak. A strong acid will never bind. It will never touch a proton, right? Because it's a strong acid. It's always dissociated. It doesn't really want to, that's what makes it strong. It doesn't want those protons. Weak acids, on the other hand, are kind of, yeah, I'll have it, okay? If the pH is right, they will just bind it, okay? So, in a mixture of a weak acid and its anion, if you add a proton, it will bind to the anion and basically disappear from the solution. And that keeps the pH stable. If, on the other hand, you take away some proton or you add some base, okay, this weak acid, because it's still acid, right, it will dissociate, it will release a proton, which will react with the base, and the pH stays the same. So buffers are no magic. They are just two components which both can either bind, well, not both, but each can either bind a proton or release a proton, depending on the changes that you cause to the solution. Either adding a proton or adding the base that binds a proton. That's it, okay? There's nothing to it, okay? We could look at some titration curves, but I'm not sure we have a huge amount of time to look at that. But basically, if you imagine that we only had a strong acid, okay? So this would be pH, and we have a solution of strong acid, and we are adding some base to it, okay? So we start at pH, whatever, 0, who cares, okay, depending on what we have. Start at pH 0, and we keep adding more and more base, right? So the pH will basically keep rising, and then suddenly, it will rise suddenly to basically to where, so we start with completely dissociated acid, and we just kind of use up all the protons, and then we'll jump to the pH of the base, basically, and just keep rising. Okay, that's what we have. If we take a weak acid, we get a significantly different picture. What we see there, it will start kind of rising, but then we get a long plateau, and then it will jump up. And it's this plateau where we have the mixture of the weak acid and its salt. And this plateau is the thing that buffers the solution, because you can see that even though we're adding more and more base, the pH basically doesn't change at all. Okay, here it just keeps changing all the time and it jumps up. Here it starts changing a little bit, then it stays for a long time it stays the same because we are doing this process, okay, adding more and more base. It allows the remaining undissociated acid to give the proton, okay? The pH stays the same, but then of course, once we exhaust the capacity of the buffer, then it just goes to all the way up and the buffer stops working. Okay, so that's the principle of how buffers work. Now, the middle of this plateau, where the buffer works well, is equal to pKa. So the pH is equal to pKa. That's where the buffer works the best. That's where it has the biggest capacity. Now, why is that? Well, because if pH equals to pKa, the ratio of the acid to the salt is 1:1. So it's equal, okay? So that means that basically at this area, if you add or remove a proton, there is the biggest capacity to use it because the ratio is 1:1, okay? Once you start moving outside of it, it gets a little bit harder to buffer, to stabilize the pH, okay? So when pH is equal to pKa of the acid, that's where the buffer works the best. And we can use this because then, depending on which pH we want to stabilize or which pH range we want to stabilize, we will just pick an acid that has pKa in this range. So we can create different buffers depending on what we want to, which pH range we want to buffer. I know this is a little quicker than than maybe you expected, but hopefully this makes a little bit of sense. Let me just add a couple more things, okay? 2 more minutes. One is, how do we calculate the pH of a buffer? For this, we use an equation called Henderson-Hasselbalch equation, and we heard about Lorentz-Henderson in the first lecture. Some of you may recall that, okay? So the Henderson-Hasselbalch equation is for calculating the pH of a buffer. And it's an important equation, so Hasselbalch equation. And you should really know this equation. Maybe you already know it. So can somebody tell me what it is? Is it plus? Well, it depends, yeah. But let's go with plus. Correct? So these are the two components of the buffer, okay? We take the ratio of those components, take a log, and we can calculate the pH of a buffer, okay? Very important equation. We'll use it even in clinical medicine. It is used, so it's an important thing. The final thing in the final minute is, what are the major buffering systems in our body? So in the extracellular fluid, we're coming back to the beginning. In the extracellular fluid, the major buffer system, or one of the most important physiologically, is bicarbonate. And bicarbonate is composed of two components. Sorry, bicarbonate buffer is made of two components. One is carbon dioxide, that's the acid, and bicarbonate, which is the salt. Now, it may seem weird saying that carbon dioxide is the acid. Well, traditionally, they would tell you that there is carbonic acid is the acid. But carbonic acid, basically, it's so unstable that it basically doesn't exist, okay? It immediately decomposes into bicarbonate or to carbon dioxide. So from my point of view, I think it's better to write carbon dioxide and bicarbonate, because carbonic acid, not really, okay? It exists for like a nanosecond or something. It's not really there, okay? So that's bicarbonate. Why is it important? It's not that important because there is so much of that. If you go back to the composition of the fluids, you'll see that there actually isn't a huge amount of bicarbonate. But the important thing about bicarbonate buffer is that we can regulate easily the composition of the buffer by breathing. Because if we breathe more, if we ventilate more, we get rid of carbon dioxide, and we change the composition, the ratio of the components. And if we decrease our ventilation rate, which we can't really do very much, but we can do it a little bit, we can move the components to the other way around. So physiologically, bicarbonate buffer is one of the most important buffers because we can relatively easily manipulate it, and it allows our body to kind of change or react to changes in pH. Both intracellularly and extracellularly, probably the second biggest, most important buffer are proteins, okay? Lots of proteins. Again, once you look at the composition, we have lots of proteins, both intracellularly and intravascularly. Okay, lots of proteins. They are very important buffers. The third important buffer, again, both intracellularly and to some extent extracellularly, are all those phosphates. Not just the inorganic phosphates, but especially the organic phosphates. Again, they have hydrolyzable groups, right? They can accept protons or release protons, so they are also an important buffer. The last buffer system that I want to mention is ammonia buffer, which is mostly present in the urine, and it allows the very efficient excretion of protons into the urine, because once again, as I said, most of our body is producing acids. So our metabolism is mostly producing acids, so we need to get rid of them. And this ammonia buffer in the urine helps us pull those acids into the urine so that we can excrete them. All right. Sorry about taking more of your time. Any questions now? No? All right. Okay. Yeah, you're welcome.